last several years, we have been developing sensory perceptual metrics as a way to determine if the brain is healthy or not. The design of these protocols is based on many more years of fundamental neuroscience research from both our work and the work of many others around the world. One of the biggest problems in assessments of neurological deficits is obtaining quantitative and biologically based measures of brain function or dysfunction. Imaging techniques such as MRI have proven to be very useful for detecting aberrations that are caused by severe trauma, stroke, or tumor. Inherent problems with these techniques, however, are that they are cumbersome, costly, and limited in resolving power. For example, modern imaging techniques are not capable of detecting differences in brain function that are caused by concussion. Additionally, these imaging techniques are also not capable of detecting acute pharmacological changes or subtle changes in connectivity that might occur within very short distances within the brain. One possible solution to the limitations of imaging is measuring the correlating perceptual changes. For example, if we stimulate a single point on the fingertip, you can detect that a region in the brain has become active. You can detect this both perceptually and with imaging techniques. If two adjacent fingertips are stimulated, two adjacent sites in the brain become active. If one stimulus is larger than the other, then one of the brain regions will be more vigorously activated than the other. The difference in intensity between the two fingertips can be easily distinguished perceptually, and this correlates well with the differences that we have been able to detect at high resolution in our animal studies that are resolved at micron resolution. However, these intensity differences are not detectable at the millimeter resolution, which is the scale of modern imaging techniques. One question that people often ask is why use the skin as a medium to test brain function? There are several reasons. First, there is a direct relationship between the skin and the brain. It is topographically organized, meaning that the skin on adjacent fingertips maps to adjacent regions in the brain. This makes it easy to design tests that look at interactions between two cortical regions. Second, it is very easy to limit the other skin inputs to the brain. If the tests were conducted visually, many parts of the brain that are associated with vision would be activated, rather than most of the activity being at two distinct adjacent places. Third, the part of the brain that processes skin inputs is highly integrated with the pain system. No other sensory system has pain receptors, and a patient's pain often has a significant impact on brain function. How that pain alters central information processing is often a very important part of a patient's diagnosis. Some sensory perceptual metrics are very robust as long as the subject is healthy. Amplitude discrimination is the metric that we use to observe how well a subject can tell the difference between two stimuli delivered to two adjacent fingertips. The smaller the number, the better they are at detecting this difference. These measures stay relatively constant across the age spectrum. Thresholds, on the other hand, are measures of skin sensitivity and do change with age. Smaller numbers reflect greater sensitivity. Amplitude discrimination, or the cortical metric that is described here, is more robust across the age spectrum because it primarily reflects how well the adjacent activated areas in the brain interact. So how can a cortical metric, like amplitude discrimination, be a measure of brain function? Here we have a picture of a brain slice that experimentally mimics two skin site stimulation. The X's mark the regions of the brain slice that are stimulated. The graph below is a histogram that shows how well you can tell the difference between the two different brain sites. If a certain chemical is put on the brain slice, the slice gets worse at telling the difference between the two stimuli. This experiment mimics a systemic decrease in GABA levels, which is common in many neurological deficits. Thus, we would predict that a number of people with certain neurological conditions would be worse at performing this particular task. This is in fact the case, as this graph depicts. Remember that lower numbers for this metric are better. In the cases of autism, fibromyalgia, migraine, concussion, and alcohol dependency, this metric is significantly higher. It is important to remember that this is only one of the measures that can be obtained. The best way to fully characterize brain function via sensory perceptual metrics is with multiple sensory perceptual metrics. Multiple sensory perceptual metrics can be assimilated into one mathematical analysis. In the data displayed here, concussed versus non-concussed subjects are compared in multiple dimensions with a method called principal component analysis. 5 tests were used to characterize each individual's brain health, and each dot or ball represents a composite of the metrics obtained for one individual. This type of analysis clearly differentiates these two populations. Note that concussion, or mild TBI, is something that cannot be detected by any modern imaging techniques. Tests and analytical methods can be optimized for any of a number of patient populations, and this process of defining or characterizing an individual's brain health by sensory perceptual metrics could prove to be an extremely reliable, efficient, and cost-effective method for assessing the impact of any neurological disorder that systemically alters brain function. Visit us on the web at corticalmetrics.com.